All right, welcome to our second video for Chapter 13. Here I'm going to walk you through a specific example of unlinked and linked genes in our fruit fly model, the Drosophila. Specifically, we're going to look at body color and wing size. Before we jump in, let's look at the symbols that are used for fruit flies. So for fruit flies, the letters that we choose are actually based on the mutant phenotype. So if I look at the symbol here, I see B and VG. So B is black body because black bodies are the mutant phenotype. They're not as common. The wild type for fruit flies, the more common phenotype is gray bodies, which is indicated by a B with a superscript plus sign. And then for fruit flies, the mutant phenotype for wing size is vestigial, which is like a small wing size. And the wild type is normal wing size, which is indicated by VG+. So here I see that this fruit fly is dihybrid, is heterozygous for both. So I can see it's B plus B, so that's gray body, because the wild type is dominant over the mutant phenotype. And VG, so VG plus VG shows me that it has normal wings. So we're going to look at 3 hypothetical inheritance patterns for body color and wing size. In our first pattern, or hypothetical pattern, we're going to assume that the genes for body color and wing size are on different chromosomes, so they're going to independently assort. So I can see that this is my original fly. This would probably be the F1 hybrid organism. And I see that body color and wing size are on 2 different chromosomes. If that's the case, I would expect independent assortment, or B plus could go with VG plus during meiosis 1, or B could go with VG, or B plus could go with VG, and B could go with VG plus. So these are my chances of producing 1, 2, 3, 4 different gametes, and this would be a 25% chance of generating each type of gamete. So I have the gametes located here. And then what they're going to do, what they're performing here, is a test cross. So I'm mating this fruit fly with a tester. So remember, a tester is always homozygous recessive for the traits. So that would have been the fruit fly tester's genotype. And here on the Punnett square, they're just showing you the gamete that's made from this tester. And if we cross these two, these are the possible genotypes and phenotypes of their offspring. And underneath I see a few notes. So I'd have a 25% chance of getting each of these 4 types of offspring. And some of these they call parental, but others they call recombinant. What does that mean? So the parental types, these genotypes are reflective or are identical to the parent genotypes if you look at these two. The recombinant genotypes of these offspring right here, these do not reflect the original genotype of the parents. So we call these recombinant fruit flies. Now let's look at our second scenario. In this scenario, the genes are on the same chromosome, but no crossing over occurs. So here I see I have my same fruit fly, but this time the gene for body color and wing size are on the same chromosome. It looks like one is located up here and the other gene is located down here. If there is no crossing over, I only produce 2 types of gametes from this fruit fly. So these are the only possible gametes that I would produce. I would not be able to produce this type of gamete nor this type of gamete. So if I perform the same test cross with the same tester that I did in the previous slide, then I would see 50-50 over here, 50% of each types of parental types, but no recombinants. Now let's look at our third scenario where the genes are on the same chromosome again, but this time crossing over does occur all of the time, 100% of the time. So again, I have that same heterozygous fruit fly. but this time crossing over is happening, even though the genes are on the same chromosome. Looks like they're far apart enough to cross over. Whenever that happens, I do get, again, the 4 different possible gametes, just like I did in my first scenario, scenario A. In that case, I can see my 4 gametes here, and then I'm performing the same test process earlier. And again, I have a 25% chance of getting each of these fruit flies. Half of them reflect the parental types, meaning they have the same genotype as the parents. And half of them are recombinant genotypes, so they do not match the parents in terms of their genotype. So in the parental chromosomes, which we really derive from the parental generation, assuming that the original parents had the wild type alleles, both located on one chromosome. And of course, this one is the identical sister chromatid. So that would be that set of parental chromosomes. And then for the other parent would have the mutant alleles on the same chromosome for the other parental chromosomes. After crossing over between the non-sister chromatids, you generate your recombinant chromosomes, which you did not previously have. So if 2 genes are far enough apart from one another, then they will cross over about half of the time. So you'll get a 25% ratio of each type. But if the genes are close together, then they might not cross over as frequently, and you'll get a lower probability of generating these recombinant chromosome types. So when Thomas Hunt Morgan actually performed this cross between that heterozygous, that dihybrid fruit fly, and this tester fruit fly, this is what he actually found in terms of the number of fruit flies for each type. So when he looked at the offspring, he found these were the numbers of parental type offspring produced, and these were the number of recombinant type of offspring produced. So when you look at something like this, and I want to figure out what actual percentage of all of the fruit flies are recombinant, what you can do is add the numbers together. So if I want to find the percent recombinant offspring, and here they tell me it's 17%, how did I get that 17%? So you want to add these two. So 206 plus 185, that gives me 391 over the total offspring. So if I add all 4 of these types, I would get 2,300. And then I want to times by 100 because I'm getting a percentage. That will give you 17%. So 17% of the offspring are the recombinant type. That's lower than expected compared to here, where crossing over is always happening. So what this tells me is that the 2 genes are on the same chromosome, but they're located closer together rather than further apart. They're only crossing over 17% of the time. Now that we know how to calculate something like recombination frequency, we've been able to create genetic maps. So we actually know all of the genes on Drosophila, on the Drosophila chromosomes, because of tests like the one we just saw previously. And in fact, one of Morgan's students, Sturtevant, realized that he could use these recombination frequencies to generate a map of all of the relative positions of the fruit fly genes on their chromosomes. So the calculation we did previously, that 17% from the previous slide, is known as the recombination frequency. And we use this number to act as a function of distance between different genes. So in our class, recombination frequency can be expressed as a percentage, just like we did on the previous slide, like that. But we're going to say that 1% recombination frequency is the same thing as 1 genetic map unit, which is also called the centimorgan. So we can also consider recombination frequency as a unit of distance. And if we do that, we would change this to 17 centimorgans. So here are some examples of the relative positions of these different genes. So for example, if I looked at body color and cinnabar eyes, it looks like they are 9 centimorgans apart. That means if you were to cross these fruit flies, you would get a 9% recombination frequency. So as I mentioned earlier, the further apart two genes are, the higher the probability that a crossover will occur. So if I have one chromosome, so let's say something like this, and I'm looking at gene here and a gene here, and I'm just going to draw this chromosome linearly instead of that X shape, and let's say it's over here and over here, and I contrast that with another set of genes. Maybe I'm looking at this gene, and then the second gene is over here, so that's gene 1, that's gene 2, gene 1, gene 3. The closer they are together, the less likely they're going to recombine. So maybe the recombination frequency between 1 and 2 is like, let's say 12%. That means they're 12 centimorgans apart, whereas let's say 1 and 3 is like 30% recombination frequency. So that's 30 centimorgans apart. The greater distance, that shows you how much more likely they're going to recombine. If they're 50 centimorgans apart or greater, say let's say from 1 to 4, let's say that's 50 or greater, then the recombination frequency is just like if the genes were not linked. If they were on 2 separate chromosomes, you would get a 50% chance of recombination every time. So again, a linkage map is a genetic map of chromosomes based on the recombination frequencies that were generated from these types of experiments. And we used recombination frequency, centimorgan. You can also express these units as map units. So we've seen all three, and it really depends on what kind of book you're using. These tell us relative distance and the order of the genes, but it doesn't tell us exactly where the genes are. We're going to be doing that more closer to the end of the semester. So again, here's another look at the picture I showed you earlier. So here we had the parental types of gametes during this cross with a tester. And then here I see some crossing over to generate my recombinants. I received a recombination frequency of 17. And how I did that was, again, adding the number of recombinants over the total number of offspring, which I put in the denominator. So that gave me 17%. So another way to look at this is the gene for body color and the gene for vestigial wing or wing size. They are 17 centimorgans or map units apart from one another. So let's review that cutoff for linked versus unlinked genes and how often they will recombine or cross over. So genes that are close together on the same chromosome, let's say those two genes, will not recombine very frequently. But genes that are far apart will recombine more frequently, like this one. And again, you know the recombination frequency based on how far apart they are. So if this was 38, they would recombine 38% of the time. So this is 38 map units, for example, or centimorgans, whereas this one is like 5% recombination frequency, which is the same as 5 map units or 5 centimorgans. But if you have two genes that are 50 map units apart or greater, or centimorgans you could say, then even though they are linked, remember linked means they are on the same chromosome, they behave as if they were unlinked, and that means they're on 2 different chromosomes. So you would receive or get the same recombination frequency that you would get if they were not on the same chromosome. So again, one more time, 2 genes that are 50 map units apart or greater even though they're on the same chromosome, they will behave as they were unlinked, as if they were on two different chromosomes, and they will assort independently. One of the greatest benefits of using recombination frequency values is to generate a genetic map, just like the one we have shown here for Drosophila. This looks like it's chromosome number 2, and we can see the relative distances of each of these genes from one another. And these numbers were generated by looking at the recombination frequencies of the offspring, the number of recombinants that we had by mating these different fruit flies. So we're going to be practicing doing something similar using recombination frequencies to generate a genetic map towards the end of this week and the beginning of next week. And we'll see some more practice problems in either the lab and or lecture homework. All right, that takes us to the end of our second video. In our third and final video, we're going to look at abnormal chromosomes. How do we get an abnormal number of chromosomes? And also, what are the effects of these abnormal numbers?